Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base Biotech podcast, and this is number 16, I think, going out on the 4th of July. Happy 4th if you're in the US, and happy belated Canada Day for July the 1st as well. And in case I forget, happy Caesar Salad Day. It's also Comic Sans Font Day. Less said about that, the better. We're managing to keep up with more biotech news articles on deeptechdigest.com. Having been in the media for several decades now, this is one of the worst times of the year for news. The summer, which is called silly season, when you run silly stories that you'd not touch at other times. And the other bad time is the week after Christmas because nothing's happening. But if you do have news or suggestions for who should be on the podcast, or indeed any other suggestions or comments, please do get in touch at jim@deeptechdigest.com, which is also the address that you can use. Which is also the address you can use if you're interested in sponsoring one or more podcasts or advertising with us. Last week our guests were Shai Meltzer, head of Israel's National Bioconvergence Programme, talking about, as you would guess, bioconvergence, and Phil L'Huillier, the CEO of Scancell, talking about cancer vaccines. This week it's a bumper week because we have 3 conversations for you. Maybe you can listen over the long 4th of July weekend. We talk to Sean MacDonald, CEO of Domain Therapeutics, Carina Kern, CEO and founder of LinkGevity, and Nadia Harhen, General Manager of AI Simulation at SandboxAQ. Now that we're carrying more news, let's recap some of the headlines of the biotech news over at deeptechdigest.com. Hancor Bio presented promising data on its checkpoint inhibitor. Enterome had positive interim results in relapsed refractory indolent non-Hodgkin lymphoma, and a new collaboration between Thermo Fisher Scientific and Cellular Origins looks to deliver affordable CGTs to patients. Angel Yeast is opening a new biotechnology park. Innovent Biologics showcased its dual innovations, and 3 companies have worked together to release a 30 million cell atlas. UCB announced positive CDKL5 deficiency disorder study results. CMUH and Shine On Biomedical are looking to push Taiwan into the exosome-based precision medicine space. And Rocket Pharmaceuticals received FDA IND clearance for BAG3-associated dilated cardiomyopathy treatment. ProBio opened its plasmid and viral vector manufacturing facility. AbbVie is acquiring Capstan Therapeutics, and Oxeltis is raising funds to develop an antiviral for Bunya virus. And you can read all of these and quite a lot more at deeptechdigest.com. Right, as we've got a lot to squeeze in today, let's chat and let's get things moving with an interview with Sean MacDonald, CEO of Domain Therapeutics, a company delivering breakthrough GPCR-targeting therapies in immuno-oncology and inflammation. So I guess first, if you could give me a little bit of background on Domain Therapeutics.
Sure. So Domain Therapeutics, it's a GPCR drug discovery company. It actually got its start almost 20 years ago. And with relatively little dilutive financing, it's managed to survive for the majority of that time without much in terms of investment by VCs. And it really did that as a result of the strength of its platform. And so it entered into a series of different pharmaceutical collaborations, created new codes that eventually got bought, and brought in over 80 million non-dilutive financing during that period as a result of the quality of its science and the quality of its platform. 2021 took the decision to kind of follow in the footsteps of more traditional European companies that have made the leap from platform to global international company. You can think of Ablinks, Morphosis, Argenx, et cetera, where they start with platform deals to validate and then move into their own intrinsic value creation by building up their own pipeline. And that was around 2021 that they did that, closed the Series A, initially focused just on immuno-oncology. In 2024, we expanded that out to focus as well on inflammation. And so we now have a pipeline that's reflective of both those therapeutic areas, still leveraging the platform, but now in the process of just closing our Series B to take us through the next couple of years, where we've got a series of different catalysts associated with our pipeline that we're hoping will really draw the attention of the crossovers in the future. And potentially, we have an eye on public markets out towards 2028, 2029.
Pretty busy for the next little while then.
Yeah, it's been interesting market conditions, to be able to do all that. I think we really have the benefit of that history and that track record going for us in the discussions that we have. And now, operationally speaking, I think that as we move towards the clinic, the milestones that we're delivering on is what's catching people's attention. And I think that's been extremely helpful to secure that. But to your point, you don't get to rest very many days in between the last financing and the next financing.
No, absolutely. I've seen the term used with regards to your company redefining cancer therapy. I wonder how or if you could tell me how that is the case.
Sure. So GPCR is a very, very well-known drug class. I think you could equally look in press from 20 years ago and today, it remains interesting. And one of the reasons is because there are so many, and it's involved in so many different biological processes. So I think that historically, the GPCR drugs or the drugs against GPCRs that have been developed is really more on the cardiometabolic side as well as in neurology. And so the fact that we're looking specifically in the tumor microenvironment for GPCRs of relevance to the disease biology of cancer is really the added value that we're bringing. And so not a lot of companies, as a matter of fact, there are very, very few companies that are targeting GPCRs that are relevant for cancer. This is a different approach to immuno-oncology. And so a lot of immuno-oncology drug development is focused on how it is that we can kind of break that therapeutic ceiling for the current standards of care. And we're doing the same thing. But the approach that we're taking is very, very different, focusing on this very, very interesting class and highly druggable class of GPCR targets and leveraging a technology that has proven itself to really be able to, I would say, parse out the appropriate downstream signaling of exactly what it is that we want to hit, and therefore exactly what it is that we want to contribute to the overall combination therapy.
Is anybody else in the field doing that kind of thing, or is what you're doing unique?
Our strategic competitors, so we're not the only company targeting GPCRs, but if you take a look at most of our strategic competitors, they're in cardiometabolic, they're in kind of the more traditional, I would say, areas of GPCR drug discovery. Part of that as well, the platform itself lends it to being a little bit more, I would say, applicable outside of those traditional areas as a result of the fact that we're focused more on functional signaling of the native GPCR rather than trying to lock it into place. And that allows us to work on other types of targets, including orphan GPCRs and things like that, which the others are a little bit more limited by.
Could we move on to maybe your programs, and if you could tell me a bit about the DT9081 oncology program?
Yeah. So our DT9081 is a small molecule antagonist of the EP4 receptor. And the EP4 receptor is part of an inflammatory pathway that actually starts with COX-2 and then moves through prostaglandins, in this case PGE2, and then finishes off with EP4 receptor signaling. It's been validated, and so there is actually lots of survival data that if you do block COX-2, it does have a positive influence on survival in cancer. However, if you block at the top of the cascade, then you end up with some undesirable side effects because you're blocking more than just PGE2 signaling. You're blocking other prostaglandins as well that can have some different toxicities. At the level of EP4 receptor, however, so at the level of signaling rather than at the level of initiation, then you're able to still benefit from, the hypothesis is we're able to still benefit from that survival, that additional survival, but, minimize those side effects. And so it just completed a phase 1A monotherapy dose escalation at the end of last year. At the moment, we're just finishing off. We should have the full report in the next few months and getting it ready to move into a sarcoma trial, which is going to be in combination with immune checkpoint inhibitor and chemotherapy in 2026.
All right. And what about the DT7012 program, and maybe where's that at?
Yeah, so the DT7012 program is a very exciting program for us. So it's actually a monoclonal antibody, and it targets CCR8. Now, what makes CCR8 particularly exciting is that there's a tremendous amount of competition. So it makes it kind of scary as well. But I think the amount of interest in this target is pretty impressive. Several large pharma are also developing antibodies against this target. And that's really because there has been over the years a profound interest in Treg depletion as a strategy for really amplifying, I would say, the response to current therapies, but also to reinitiating that immune response within the tumor microenvironment. And I think some of the past approaches, even recently things like CCR4, have not really delivered for a variety of different reasons, but one of which, the hypothesis being that there's still too many Tregs outside of the TME expressing that target to be able to really get the right therapeutic window, particularly in solid tumors. What's different about CCR8 is this is very highly and specifically expressed on tumor-infiltrating Tregs, which is what's generated the tremendous interest. And so we have an antibody that follows the same mechanism of action. It's an ADCC-ADCP optimized. So what that means is it's been optimized to be able to interact with the immune system to facilitate then Treg depletion within the TME. And also, too, we leveraged our platform to develop a profile that differentiates it. And this was recently presented at AACR, the preclinical differentiation that we have on two main fronts. One, CCR8 is a post-translationally modified target. We bind to all the different post-translational modifications, which means that we have a broader applicability. So there's no escape mutants that can come about and escape our product. And the second, very importantly, is we actually block internalization of the CCR8. Now, this is very important, as I'm sure you can appreciate. In order to be there for the Treg depletion, you need to be on the surface of the cell. And as a result, us and BMS and a couple of others weakly block internalization. It's really us and BMS that block it, and that has a direct impact then on ADCC activity in the presence of high concentrations of the CCR8 ligand.
You mentioned AARP. What was the presentation there, and how did that go down?
Great response. I think there's an argument to be made that 2025 is going to be a big year for CCR8 because some of the larger players are going to be releasing data. So there's a lot of interest around the target. I think that we had presented at SITC last year as well some of the first outputs of this differentiation data. This is when we started releasing who we were benchmarking against. And so I think a really good response and lots of questions, and particularly relevant because we are opening sites as we speak to get ready to start our clinical trial in the next few weeks here. So I think the response was good, lots of interest around CCR8 generally, and specifically to our differentiation data. This is something that people are pretty excited about to see how it pans out in the clinic.
I mean, obviously AACR is a really big one. Do you go to a lot of events to talk about your work, and is that an important part of what you do?
It is. The team is getting ready to head to ASCO tomorrow. So definitely on the oncology side, I think that we have been looking at ASH as well, as we have some hematology applications that we're exploring, as well as some collaborations more on the hematology side. I would say AACR is an important one. SITC is an important one, on the immunotherapy, more preclinical type of data that we're generating around our different assets. But ASCO, as we move deeper into the clinic, is extremely important. Great opportunities to meet with key KOLs, investigators, build up those networks. And more, I would say also on the corporate side, it's an opportunity for business development. So before ACR now, they have really a partnering day or a couple of, an opportunity to meet with the big pharmas, understand what their development strategy is, understand what they're looking for, and then engage with them in a structured setting rather than historically where you really have to send out a bunch of emails and manage your own calendar. And so they've recognized that it's an opportunity beyond just the science, which as a CEO is a great return on investment for the participating in those types of meetings. Could you tell us a little bit about the preclinical insights on the PAR2 biased NAM program and the relevance of that? Sure. So PAR2 program is another great example of how the platform helps us deliver unique profiles. And so an antagonist that blocks the PAR2 target will block both the G protein signaling as well as the beta-arrestin signaling. And in this particular case, you don't want to block internalization, because what ends up happening is that then there's the potential for more PAR2 then being expressed or compensating expression at the surface of the cell. So in our particular case, we are an allosteric modulator and biased to only block G protein signaling, not beta-arrestin signaling. So we don't actually, in this case, block internalization, but we do block the signaling pathway. And that is very, very different from the rest of the field that are mainly antagonists and mainly biologics. PAR2 is a target that people have been looking at for a long time. A lot of big pharmas have tried and been unsuccessful in developing a small molecule targeting PAR2. It's a testament to the program that we were able to kind of crack this tough GPCR target. But what's great about that is there's a large body of academic literature, as well as others that have tried to develop PAR2 targeting molecules, that shows that it's expressed on a variety of different cell types and therefore has a potential broad activity on pain, on barrier integrity, fibrosis, as well as Th2 inflammation. And it's the Th2 inflammation part that makes it particularly interesting, because that gives it both an applicability within the tumor microenvironment, where Th2 inflammation can actually drive cancers forward, as well as in an inflammatory environment where blocking Th2 inflammation is important anti-inflammatory effect, particularly against some of the others, it's more Th1-driven inflammation. And so for us, the preclinical data that we showed at ACR was its applicability in immuno-oncology in collaboration with Professor John Stagg here in Montreal. And he demonstrated in some different models that bringing our molecule that targets PAR2 into combination therapy has a very, very important additive effect of standard of care cancer therapy. We're quite excited by it, because it does continue to flesh out or round out the package, the very, very interesting package we already have in inflammatory disease for that molecule.
So where does that go from here?
Great question. I think that we are moving this forward in inflammatory disease. As a matter of fact, we just completed a couple of advisory boards in order to position this clinically. The PAR2 program is going to be moving into a clinical trial in early 2026. So we're just finishing off the final toxicology through the back half of this year, and CMC getting ready for that trial start. It was a tough choice because the data in oncology is very interesting. I think that what kind of tipped it in favor of inflammatory disease is that we have the potential both as a strong monotherapy as well as in combination. And I think that ultimately we felt that from a balanced portfolio perspective, given that CCR8 as well as the EP4 receptor program are both in immuno-oncology, having this as a mitigating offset to being too concentrated in one therapeutic area was also a sound strategy.
When it comes to strategies like that, and you have lots of different things going on, when you kind of develop something else, how do you deal with the fact that you're adding one more thing to the portfolio, and like the manpower and everything that's associated with that?
Yeah, it's a great question. It's one we struggle with. I joked earlier about some of the challenging market conditions at the moment. It means that I would say resource constraint is even more of a consideration than under normal circumstances. We have to be really tight with our storytelling, which means we need to be very disciplined with our investment choices. I think, however, we still want to do our best to maximize optionality. It's drug development. We don't know what's going to work. And just like we don't want to put all of our eggs into a single VC basket, we want to diversify, talk to different investors. We want to have business development opportunities. We want to have other types of, whether it's grant opportunities or collaboration opportunities to move these programs forward. On the scientific side, we also have to have a good idea of how our molecule is working. The reality is that even if we do position inflammatory disease, these extra applications give us a better depth of understanding of how our molecule is working, and can ultimately allow us to position better or position slightly differently than we would have otherwise. But it is something where, unfortunately, we do have to operate in the resource-constrained environment and stay disciplined, that, we utilize this data to move forward, sometimes just supporting the original strategy, maybe tweak it somewhat. But it's rare that we can then sort of launch a whole parallel set of trials, based upon this type of data, unfortunately.
We do hear about things like artificial intelligence, machine learning, being able to shrink timelines and help. Is that a consideration, or is that just not really a part of the equation?
I would love to have an AI piece to our company right now, because I think that the frenzy around AI investment, and I would say the open-mindedness to trying all sorts of different AI type of programs, is definitely there. It's tangible. We want to call it computational biology, AI. There's a lot where they're trying to merge with IT. A very striking comment at a recent conference for me was somebody said, the only thing that we can say for sure about AI is that the system you're using right now is the least sophisticated and worst one you'll ever use because it just gets better every single day. I think the other striking comment was around the data sets that they have access to in order to actually come up with their answers. And so right now, and it's part of the reason or part of the debate around big pharma sharing negative results from old trials and things like that, because those negative results actually become a competitive advantage for them based on the data sets they then have to train their internal AIs and how they can then ultimately use that for their own internal drug discovery versus a biotech that needs to rely probably more on a vendor unless they are an AI company themselves that have access to maybe proprietary data sets from universities or other groups. So for Domain, we struggled to really identify, one, the right AI that can really add more value beyond what it is we already have from our platform. We remain open and we have active discussions ongoing with a couple of different vendors. And so we are trying to figure out how to best integrate this. But it seems, at least for us at the moment, that there isn't a CRO collaborator that can really replace the hard, laborious work of drug discovery, validation. There's unfortunately no IT solution to skip the tox study. It still needs to be done. We still need to generate the data set and, have that ready for regulators. So absolutely, it's something that keeps me up at night because, it's becoming more and more important. It's a question that's on the tip of the tongue for a lot of VCs right now. Like, how are you guys thinking about it? But we just haven't identified something that can really move the needle for us, whether it's in terms of cost savings or time savings, based upon what's available out there.
With the existing programs that you have, what does success look like, or what are the goals when it comes to the end patient?
I'll start with the CCRA program DT7012. As we look ahead to the next 9 to 12 months, we expect to generate a biomarker proof of concept in patients for Treg depletion. And Treg depletion is kind of a recognized endpoint. And for us, that's one and very important catalyst that's going to be arriving in 2026. We're also going to be kicking off in 2026 some indication-specific cohorts and expect data from that towards the end of 2027. And so we'll have a combination of Treg depletion, proof of concept data, as well as then actual clinical outcomes within a specific indication by the beginning of 2027. So for us, success is really being able to move that forward, hit those timelines. Those are two key catalysts, and then ultimately trigger a financing around those key catalysts to then be able to drive us forward. What we hope is going to then be a pivotal trial that will be able to ultimately launch this asset before the end of the decade, so around 2029 timeframe. On the part two side of things, I think that there are three different, I mentioned part two is expressed on a variety of different cell types. So it has a triple mechanism of action in inflammatory disease. It hits Th2 inflammation, as we talked about, so standard anti-inflammatory, but also it is expressed on epithelial cells in the gut, keratinocytes in the skin. So it has an impact on barrier integrity. And then lastly, it's expressed on DRG neurons, so also on the pain side of things. And we're really looking in our initial proof of concept of patients to try as best as possible through a series of secondary endpoints to show the impact on these additional aspects of the modality to position ourselves well to be able to combine. And, initially we're looking at a couple of gastroenterology indications where we could combine them with some of the standards of care that are out there and really add something beyond what it is they have right now. So as I mentioned, we just completed a couple of medical advisory boards. And so the patient portion of our trial is going to kick off in Q3 of 2026, and we were hoping for that data in Q3 of 2027. And so that's what good looks like for the part two over the next couple of years. I think we remain opportunistic for both assets in striking the right partnership at the right time, but ultimately, I think that we have a good plan. We have good investor support at the moment, and so we're hoping to be able to hit those catalysts and execute well.
As if you weren't busy enough, are there any other things that you're looking to add or to different conditions that you're hoping to tackle?
So I think from a therapeutic area standpoint, we're going to remain focused on IO and inflammation. Both offer up plenty of opportunities for our platform and for our drug discovery efforts. The tough part is really deciding what comes next. So we have a good research group. We've maintained a research function at the organization. It's very strategic for us the platform that we have. We have the opportunity to enter into platform collaborations in therapeutic areas outside of inflammation and oncology, as well as on a select basis inside those two therapeutic areas. We have other targets that are ready for further development. And so, as I mentioned, Domain, the platform itself has been very, very productive, and there was more than we could get financing for coming back to that resource-constrained environment. But as we move forward now with this next financing, there's a couple of other programs that we're looking to drive forward. One kind of inflammatory fibrosis and another in inflammation. But also, two, there's a third opportunity in immuno-oncology that we would love to be able to bring forward. It's more resource constraints than a lack of opportunities that the platform provides. For us, optionality, it's a core part of our philosophy. Optionality for our shareholders, optionality for our drug development. What I'm fond of saying is that we're in a great position right now with respect to a potential financing coming together, and everyone's really happy about it. But that was plan B and C. Plan A didn't work out. And if we didn't have already plan B and C ready to go forward, then we would have been in a very, very different position today than we are now.
All right. Those are the only questions I had. Is there anything that we didn't talk about that you wanted to mention?
Domain has been around for a while. I think that it's been successful as a result of quality science. It's been successful as a result of a strong technology platform. With the strategic pivot that happened in 2021, we're kind of marching along that same pathway other European biotechs have taken. And as you can probably pick up from the accent where I'm located, I'm part of that North American pivot. And I think the next stage of Domain's development as we drive further into the clinic, as well as start to expand further out in North America, positions us very, very nicely for future growth. And I think, over the next few months, we're very hopeful with the closing of this financing that we're going to be one of the few survivors of the current market turbulence, but, still set on a very, very favorable path moving forward. So I think great to have the opportunity to have these types of discussions. Very appreciative of the time, and thank you very much.
That is one busy company. Lots in the pipeline there for sure. A bit like this podcast. Next up in our pipeline is SandboxAQ, which recently raised $150 million from top investors, including Google. It's pioneering the fusion of artificial intelligence and quantum to accelerate breakthroughs in many fields, including biopharma and biosimulation. To tell us about the company and how it's helping biotech and biopharma is SandboxAQ General Manager of AI Simulation, Nadia Harhen. First then, I wonder if you could tell me a bit about the history of the company and some of the areas that you work in.
Yeah, absolutely. So SandboxAQ, we are a company that spun out of Google about 3 years ago. Some of the divisions that we have here incubated inside of Google for a few years before that. And so those divisions are related to cybersecurity, where basically we make algorithms that guard against the threat of encryption being hacked as a result of quantum computers. And so that is our cybersecurity division, headed up by Mark Manzano, who's my counterpart over there. And then we have two sensing divisions where we focus on using the Earth's magnetic field to help us navigate in situations where GPS is not available. So where there's GPS drift or spoofing, which happens quite often, even on commercial flights. And you imagine over war zones and things like that, it's definitely an issue. That division focuses on ensuring that planes can go from one place to the other repeatably and reliably. And then we have a medical device division, actually, which is pretty exciting. And there we focus on using the magnetic field of the heart. So the magnetic field in the body, specifically, we're starting with the heart to detect heart attacks, where it's kind of funny. We're all wearing these ECG devices on our wristwatches, but heart attacks are still the number one killer. And these devices are just not sensitive enough to be proactive and really alert us to underlying health conditions. And so we basically have a sensor that is able to pick up on very minor disturbances in the electrical signal from the heart in a diagnostic capacity, for which we're under a few different clinical trials around. That is another very exciting division. And then there's AI simulation, which is the one that I run. And AI simulation is pretty exciting because We did not exist upon spin out. We were just a few physicists, and we had like one software engineer, and in fact, our physicists were modeling black holes. And, and one day we were meeting with pharma companies, and they were telling us some of their most difficult problems within small molecule drug discovery, within late stage kind of clinical assets, wanting to de-risk the safety and efficacy. And then another day we were meeting with industrial companies within oil and gas, big consumer brands that wanted to improve their materials, that had ongoing R&D efforts. And we realized that the technology that we have is just so broadly applicable. Simulation, simulating the quantum mechanics of materials, of chemistry, of drugs, enabling them to perturbing them millions of times to get to the most optimal configurations, the most optimal properties. That was a very broad application. And so we subsequently, I spun up two different divisions. So one is our chemical simulation group, one is our biosimulation group. And now in our biosimulation group, we're in over 18 different therapeutic pipelines. We don't have our own drugs. We try to enable the companies that we work with to develop novel IP. We only own the software IP. And so that's our biopharma division, and it's an exciting time. We're in neurodegenerative diseases, different types of oncology programs, and then on the chemical simulation side, we have worked with a number of partners on the battery technology, battery innovation side, catalyst discovery. And, we're just doing these incredible things that have never been done before. Most of the time I just feel like I'm living and working in the future, which is exciting given the fact that we have, so much interesting and exciting technology that has recently come into our lives. This is even steps beyond that.
It seems like it's evolving really quickly, and you might even have two more new divisions by the end of the conversation that we're going to have.
I hope not. It's funny you say that. We actually had a financial simulation division as well, where we were looking at high dimensional optimization types of problems, and like index tracking, portfolio optimization, things like that. And I actually moved that to a different part of the company, which is our AI group within the company. It's called SAGE, with AIGE. And, so that is exciting because it's breathing new life over there. But yes, it's a bit wild.
And you're called SandboxAQ. What is the AQ part of that?
Yeah. So the A stands for AI and the Q stands for quantum. And quantum physics, but not quantum computers, because we are interested in applications right now. And so quantum computers are not, in the here and now. They're at least, 10, 12 years away by our latest estimates. And so, that is not a technology that we want to wait around for. We want to leverage supercomputers, high performance computing, large scale compute, which are very, very capable today to solve quite a large fraction of the problems that people are basically holding out for quantum computers to solve.
How can you help biotech or life science companies?
Yeah, several different ways. We could help them in small molecule drug discovery, which is a crowded space, but we have tools that don't exist anywhere else. And so the idea there is, if you've tried everything and nothing has worked, please come call us, because that tends to be where we shine. And so we saw this in the case of working with a Nobel laureate, Dr. Stanley Prusiner, where he had tried all of the available tools on the market and nothing worked for the stacking protein ensemble that he needed to disrupt to stop basically the formation and the progression of neurodegen diseases. And so that is one example of how we've helped. Any companies working on, for instance, like antibody design, biologics, that sort of thing, that is something that we have special tools for, in the same way that you have tools for small molecules, different kind of computational methods for binding affinity, different things. We have those tools available for biologics, for antibody design, and also when it comes to later stage, this is an area of active research and de-risking for us. We are actively trying to solve for many attributes of ADME and toxicology, and bringing those screening procedures up earlier into the process. So before you spend millions and sometimes billions of dollars if something is going to fail. And knowing that earlier on has obviously a ton of benefits, not just for the companies that are involved, but also for humanity.
There's a lot of companies that are looking at AI and not really knowing what it is and trying to incorporate it into what they do. Is there awareness of what you're doing with AQ, and how are you getting that message out to companies that you can help them?
Yeah, I mean, we are a new startup, right? SandboxAQ. We've been around for 3 years, but we don't have the same brand recognition of a Google, right? Even though we came from Google. And so that is something that we've been actively trying to get the word out. And so many of us spend time speaking publicly at conferences. There is a strong marketing effort to sort of build some brand awareness, particularly because, we're not established, but also we are looking to break in and basically make some cultural changes within massive industries. And so brand awareness is part of it, but also pushing the envelope, having people understand what our case studies are, what our proofs of values have been, where we've been successful. All of that just takes a tremendous amount of effort. And because it is so deeply technical, it's not as simple as running an ad campaign to basically explain, to a lay audience. It's actually quite detailed and requires a lot of contextual awareness. And the conversations we tend to have with customers, it takes 10, 15 minutes to orient someone in the science before you can even get to the punchline. And so it has proven to be challenging, but certainly, SandboxAQ.com. We have our YouTube channel with lots of different videos. Many of them are actually educational in nature, so just learning about this field. Part of our values is basically to educate the next generation so that we have workers in which we can hire from, because people might not be skilled in some of these areas in which technology is moving. And so those are definitely places you could learn more.
What kind of benefits can using AQ provide?
Yeah, so faster time to market, qualitatively better molecules that you understand the performance of, the characteristics of, the safety profile of. So a target product profile is something that we can engineer to from an in silico perspective. And so that is something that right now needs to be done in a very cyclical and serialized manner. And all of that we're bringing in silico very quickly in an iterative fashion with multi-parameter optimization. And so it's an increase in quality, it's an increase in speed to market, and it's cost reduction as well, because you're not fraught with failure and trial and error and not really understanding what's going on.
I know you said it's not been that long as a company, but what successes have you had so far?
As a company, first off, we've raised almost $1 billion collectively between our first and second round, which I think speaks to the vision of what it is we are doing here. We are here to have deep impact at scale, multi-generational impact, where we can all be proud of our work, when our grandchildren are sitting around looking at the things that we've put into society, new chemicals, materials, new types of drugs that are going to impact the world and really benefit humans that are suffering today, for which there is no cure. And so, I think from that perspective, our investors are really here for us for the long term. And I think that is just such a strong testament to what we're doing, who we have hired to get those jobs done, et cetera. My division in particular, we have dozens of customers. And so, these are real customers, big logos with actual problems that we are working to solve. Some of them are public, some of them are not. We're working on, showcasing more of those examples. But, again, I think that just speaks to the breadth of the technology and what we can do. And so, I am extremely proud to have our teams really understand their problems and develop solutions that solve those hard problems and scalable ones, right? So we can go back and solve very similar problems for other types of customers along the spectrum. And so there really is a lot to be proud of. I mentioned before we're in 18 different therapeutic pipelines. Most companies would have to develop their own drugs over the course of 10 years to achieve numbers like that. But our model allows us to not compete with our customers, but help them. And then, we are working with a number of different customers on developing new kinds of battery chemistry. We also work with the federal government. And so really, it's been very exciting to see this technology flourish in such a real way to solve customer problems.
I guess you've been doing some work with Sanofi and the Michael J. Fox Foundation. Are you able to say anything about those projects?
Yeah. For Sanofi, we are working on a drug that's later stage. So it's actually in clinical trials. And we are working to help them understand the mechanism of action so that they can more clearly explain to the regulator what is going on. And the long story short there is that, we are basically trying to help them access a broader on-label market as a result of working with us. So directly impacting the commercial landscape of that particular drug once it's approved by the FDA. So very excited about the work there. With Michael J. Fox, we actually have a number of different projects with them. And, Michael J. Fox is, of course, a strong supporter and a key member of the community as it relates to Parkinson's disease. And so it's like a real honor and a treat to work with this group to advance this particular disease area towards cures. Michael J. Fox, they have some of the most critically important and deep longitudinal data on Parkinson's disease. And so Sandbox as a whole, we kind of thrive in data sparse regimes. We don't need that data, but if the data is available and companies are happy to share it, we're able to utilize that in a much more impactful way. And so there we are looking to identify different types of drugs. We actually are working on several targets that they've already identified and trying to drug them. So exciting times ahead in, not just seeing that research come to bear, but translating that research into actual cures.
You mentioned simulation earlier. What could you tell us a little bit more about what biosimulation is and what you're doing there?
Yeah, it's literally simulating molecules atom by atom, the electronic structure relative to another molecule and that electronic structure. When you put structures that closely together, they have a way that they behave around one another to return to a ground state. And so the laws of physics and quantum mechanics in particular really define how these molecules want to behave in the presence of one another. It's much more complicated than that, but that in and of its core is effectively the types of simulations that we do to understand the binding affinity and the ground states of molecules. And then you can look at different other types of properties and, how would a molecule behave in the presence of liquid, blood, water, different attributes. If they're perturbed, molecules are not static. When they're in the body, they have to behave in the presence of all kinds of other stimulus and different types of materials. And so that is the type of thing that we simulate and try to understand, in the presence of a liver cell, is it going to bind to that and create a toxic effect? So that's the crux of our simulation work. And it's pretty cool. It's actually a visual. So when you do the simulations, you can actually see what's happening. And so it's not an indirect sort of wet lab method where you're probing the release of some enzyme that you're inferring might have had some effect upstream that resulted in that. You're literally seeing the simulation play out like a movie on the screen. And that has been really elucidating for understanding mechanisms of action for many of our customers. I distinctly remember watching Dr. Stanley Prusner when he saw the mechanism of action of the prion proteins. And he had been studying these proteins for years indirectly and trying to understand what sort of cascade sequence of events might be leading to what he knew was happening. And when he saw it play out as a movie. I mean, his mouth was open. I mean, he was just so shocked to see, gosh, after all these years, like that's what's really going on. So the beauty of simulation is that it allows you to see the series of events, of interactions, and that sequence is really the key thing. Typically, if you have something that binds and it does another thing, observations on a wet lab are sufficient because there's a direct kind of correlation. But when you have a sequence of events, you have no idea what has just happened. It's really difficult to go back and infer the logical sequence of events. So that's another way that we tend to thrive is in those really complex molecular sequence of interactions.
Obviously, we kind of touched on it earlier a little bit, but AI and quantum seems to change on almost an hourly basis. How do you stay ahead of that and incorporate those advances into what you do?
Yeah, I mean, you're right. It does change so frequently, and it is hard to stay on top of it. I really do rely on my team. We have very prolific conversations about all of the things, all of the data sets, all of the algorithms, all of the newest, latest things that are happening, being released. And so, obviously, like we're deeply in touch with the community, with all of the thought leaders. We have an arrangement ourselves with OpenFold. So basically, we try to work with the best of the best. And so we're already plugged in from that perspective. And then our people are at the cutting edge, like they are advancing the state of the art. So they're obviously aware of the state of the art as they're pushing it further and further along. And so, yes, like on a daily basis, like, they're sort of tasked with knowing what's going on and pushing the envelope as it relates, and then having that feedback loop with the rest of us so that we can input that. But yes, I mean, our customers, when they work with us, they're accessing things that no one else has been able to access. That's an incredible advantage. I think we're bandwidth constrained in that we can't work on everybody's problem that come to us. We can only pick a few, and we want to help those customers get a competitive advantage. And that's effectively what we do when people come and work with us.
Is there anything that you wanted to mention that we didn't cover?
More to the point on getting the word out, this is a new way of doing things. It is not traditional. It's, not a piece of software that we throw over the wall and ask people to use and get the results that we get. It's really a partnership. We work on a milestone basis on our customers' hardest problems. And, to the extent that there are challenges that customers have not been able to make progress on or have tried and failed, we'd really welcome an opportunity to learn about those challenges from our customers and see if there's an opportunity to partner and help those customers get a competitive advantage. And in situations where funding is limited and efficiency matters, please come talk to us. Go to SandboxAQ.com and check us out. And you can email me directly if you would like. It's Nadia at SandboxAQ.com. Very happy to hear from folks on their hardest problems. We are game to, particularly if they're high impact problems, like please come talk to us. We would love to hear about it.
Definitely a company making waves, and no doubt it will continue to do so as AI and quantum both evolve. Speaking of the future, now we're looking to biotech in space, and aging and necrosis in a wide-ranging discussion with LinkGevity CEO and founder Carina Kern.
I wonder if you could give me a bit of background on the company.
As an overview, LinkGevity is an AI-enabled drug discovery engine, and its focus is on aging and related disease. At the moment, our flagship therapeutic is a first-in-class anti-necrotic that can block tissue degeneration. To give you a flavor of the technology, it was actually selected as one of only 12 global innovations for NASA's Space Health Program. And this is due to its potential to alleviate both accelerated aging and kidney disease in astronauts. And we're on track to begin phase 2 clinical trials for it towards the end of the year.
Are there any other things that you're working on, or is this just the one clinical trial right now?
So the anti-necrotic itself has such vast potential. It's very much just trying to ensure maximum impact. And if you look at the problem of necrosis, it's been a significant hurdle in medicine overall from a range of conditions, from kidney disease to heart attack to stroke to neurodegeneration. But also outside of medicine, if you ask yourself, why is it that there's such an organ shortage? Why can't you store, for instance, your heart or lungs for more than 4 to 6 hours? And the answer there has been necrosis. Or what are some of the hurdles in growing artificial organs and tissues in the lab? And the answer there is again necrosis, where typically when you start to cluster cells, the central region gets starved of oxygen and nutrients, and you end up with a central dead region or a dead necrotic core. And so there is such potential of the solution because it hits this fundamental piece of biology to transform not only medicine, but scientific R&D. And at the moment, our focus is really trying to maximize that potential.
We talk about necrosis, and I think it's a term that's relatively well used and well known. But when it comes right down to it, how comprehensive is our knowledge of necrosis?
I find necrosis fascinating because it is such an ancient term and such an ancient process in terms of, the word comes from ancient Greek, necros, for death. And it's been heavily studied. So there are numerous publications on the molecular mechanisms that it entails. There are also numerous publications of how it promotes overall tissue degeneration. And what's often missed is necrosis isn't just an endpoint. In fact, we had an Oncogene paper just a few days ago out that delved into how necrosis isn't just this endpoint, but a driver of loss of resilience across age-related disease. And that's very much because you see these well-characterized positive feedback loops through which necrosis directly drives damage, but indirectly as well, as it brings in a poor wound healing response. So that's when you get the chronic inflammation, that's when you get the fibrosis. And so although necrosis has been so heavily characterized and its mechanisms by which it's involved in chronic disease has been so characterized, the missing gap in knowledge has always been, well, how do you block it? That has been unresolved. And in fact, it's such a powerful therapeutic target because if one could intervene, because the other mechanisms are so well characterized, there's good potential to try and get it approved as a drug for aging by the regulators.
You mentioned the paper that just came out. Could you tell us a little bit about that and some of the major findings in that paper?
Of course, it's certainly an exciting piece. It brought together a diverse set of minds across medicine and various scientific disciplines. And it had a range of authors from the European Space Agency to the Mayo Clinic to Harvard and so on. But the crux of that paper was, let's look again at necrosis. And necrosis historically has been viewed as this endpoint of tissue degeneration. This endpoint where it seems impossible to intervene because it is so chaotic. And so it has been largely untouched in terms of therapeutic opportunities and therapeutic focus. And this paper really turned that view on its head and said, no, look, necrosis represents the central node of tissue degeneration. So much so that if one were to intervene, it would redefine medicine as we know it. And so why is it that we aren't tapping into this potential? And in fact, not just aging on Earth, but also the accelerated forms of age-related decline you see in space. Because of course, astronauts, you see this accelerated degeneration. And again, the large hurdle there is, well, how do you intervene? And again, necrosis and targeting necrosis could hold the answer.
How many diseases or conditions would this be applicable to?
So the exciting point is, look, just like GLP-1s and the data you're getting with GLP-1s today, right? And these are drugs like Ozempic, the weight loss drugs many have heard of. So the data we're seeing with these drugs is they're applicable not just to type 2 diabetes or to weight loss, but suddenly you're seeing effects across neurodegeneration. You're seeing effects across liver disease and various other forms of chronic disease. And the point there is you've managed to somehow hit a central node in the aging process. And remember, aging is complex because it is often multifactorial disease. And what I mean by multifactorial disease is more than one factor is giving rise to the disease, right? This is why when it comes to something like cardiovascular disease, you hear high-fat diet is bad or smoking is a risk factor, right? They're multifactorial. But also, you have something called multimorbidity, meaning there are multiple diseases that arise simultaneously. They're somehow interlinked. So the point is, can you hit these critical nodes? Now, necrosis seems to be one such node, meaning if one were to intervene, it's not just potential across kidney disease, but also you see similar feedback loops of destruction across liver disease, neurodegeneration, cardiovascular disease. I mean, the paper was in Nature Oncogene, and so there was a strong cancer element as well.
And so how does what you're doing relate to being able to intervene and make a difference with some of these conditions?
You know, half the difficulty in biology and in medicine is identifying the right target. That's half the battle. And so I think this paper was really a leap forward because it identified and honed in on this very important potential target, this therapeutic target that had otherwise been overlooked. Now, the other side of the battle, of course, is actually developing a drug that can block this target. How is it that for decades, with all of our knowledge of molecular mechanisms, we've failed to intervene? So on the other side, at LinkGevity, we're actually developing a first-in-class anti-necrotic to block cell death, this uncontrolled, unprogrammed cell death. The way we do it is something akin to factor modeling in economics. Very simply, where you have a complex system, can you find the factors, a small number of factors, giving rise to the greatest level of change in the system or the greatest level of destruction in the system? And using that type of a process and a very novel theory of aging, we did identify certain factors, certain targets to hit within cells. And the exciting side is we're seeing up to 90% inhibition of necrosis in the lab with it. So to put that into context, if you see 100% of your cells dying from a harsh stress, so say from a lack of oxygen and nutrients, or from thermal stress, or from oxidative stress, with this drug, we're seeing 90% of those cells still alive. In some cases, it's a near complete prevention of chronic diseases, so for instance, kidney disease in our animal models. Of course, with science, you always have to follow the data. So we're still collecting more data on this drug. But yes, very exciting potential to date and looking to take that to phase 2 clinical trials.
And are you talking about stopping disease in its tracks, or is there a potential for reversal, or is it something that you need to do before disease symptoms happen?
Yeah, it's a great question. And I think in this case, there's potential for both. So yes, if you block necrosis, you're going to stop progression. But also, you have these positive feedback loops of necrosis, right? It's just caused by different things in different organs. So in the kidney, you have these positive feedback loops arising from oxidative stress, inflammation, a lack of oxygen and nutrients getting to the kidney, as well as side effects of certain toxic drugs. They're called nephrotoxic drugs. And that then drives these cascades of necrosis. Versus if you look at the liver and you look at fatty liver disease, there it is the fat itself and the lipid itself that then causes the necrosis. It's called lipotoxicity. Now, the crux of that is when you have these positive feedback loops, these self-reinforcing loops, that brings in your poor wound healing response, as I said. So you have your fibrosis, your chronic inflammation, other processes. You get damaged or senescent cells. Now, the challenge has been hitting these feedback loops right at the heart. With the anti-necrotic, for the first time, there's potential to do that. And once you do that, it means you would be able to reestablish your normal biology or your normal physiology, and thus also regenerative capacity.
So in terms of the anti-aging aspect of this, again, is that something that would be something that you would need to give to somebody early so that there is no necrosis? Or how does the aging part of this work?
Yeah, so I think there's potential for both. As a start with our clinical trials, we're looking at giving it for acute conditions. So starting with acute kidney injury. Remember that to date, no viable therapeutic has been approved for acute kidney injury because in large part, we've never been able to block necrosis, which is acute tubular necrosis. This is the endpoint of all of the stressors in the kidney. And so there's potential, yes, for this more preventative type approach. But in the longer term, we are going to develop it for your more chronic conditions with the potential, yes, to also try and reverse the disease phenotype or the disease presence.
And you already in passing mentioned the connection between this and space exploration. I wonder if you could go into a little bit more detail on the challenges of going into space and how this would help with that.
Yes, it's certainly a very exciting area. And I guess just before I say that, one reason I find it so fascinating is not just the possibilities of pushing the frontiers of science with space exploration, but also the immense utility in terms of all of the technologies you can bring back to Earth to potentially revolutionize healthcare on Earth. But on that vein, so one of my collaborators had an exciting title to a paper. It was in a Nature journal. It was called Cosmic Kidneys. Would your kidneys survive a trip to Mars? And the point there was your kidneys are one of the most, if not the most susceptible organs to stress. And so on a long duration mission, you're moving out of lower Earth orbit. You now have microgravity, cosmic radiation, compounding the stresses you would see on Earth. And so you get this accelerated degeneration, in particular of the kidney, to the point where you have to ask, would astronauts need a transplant or dialysis before they even get to Mars? And so I think the potential there of the anti-necrotic really, where it shields your cells and it shields them from these stressors and stops these feedback loops of destruction, there's vast potential not just for protection on Earth, but also for extreme environments where you see this more accelerated degeneration like in space.
And I guess in some respects, it almost brings up an ethical question of prolonging life, or people live longer. There's stresses on the planet as we are with 8 billion people and the lack of resources. So, is there an ethical aspect to this as well?
Yeah, I mean, as you can imagine, this is something I've pondered a lot being in the space. And I mean, my motivation really for going into the field in the first place was very much to do some good. And on that note, look, I always say I'm not scared of dying, but I am scared of becoming decrepit. And I am scared of all of these degenerative diseases one gets later in life that really robs one of their human dignity. And in terms of sustainability, I mean, the two sides there are one. There are many ways in which we can be sustainable. Withholding medication is not one of them. I don't see that as an option. The other side is also the cost of all of these diseases. I mean, just take kidney disease, right? The cost of dialysis, transplants, and so on. I mean, if you look at the US, kidney disease costs Medicare around 24% of its annual budget. It's around $125 billion. Europe is similar. It's around 125 billion euros, actually. So significant cost. And I mean, in terms of sustainability, alleviating these degenerative diseases is likely to do a large amount of good.
No, I wasn't suggesting that we should, stop people getting better and stop degenerative diseases. I just think that in some respects that when you're talking about something like necrosis, inevitably some people are going to interpret that as discovering the fountain of youth or simply prolonging life so that we look the same at 80 as we did when we were 30, but clearly it's not that. And I think sometimes it's necessary to limit those expectations as to what this kind of technology and what these kind of drugs can do.
I think there's vast potential to redefine health span and lifespan, but it's just how you phrase it and how we're conditioned towards it. I mean, if all we have in our heads is the fountain of youth and living forever, versus the reality is these diseases are robbing us of our health span. And of course, it's the same thing that then robs you of your lifespan. And in a way, we're conditioned to say, well, if you're taking out type 2 diabetes that kills you, or cardiovascular disease or tuberculosis, that's fine. But for some reason, aging, which is linked to all of these, isn't fine, right? It's quite ironic, I find. But again, it's just how you phrase it, I think. And you hit the nail on the head there.
Could you tell me a bit more about the clinical trials that you're doing?
Sure. The way I've tried to focus these clinical trials, and you will see there's no lack of ambition when it comes to our team, is how do you aim this for maximum benefit? Now, I chose kidney disease because, one, it's an unmet medical need, right? There are no viable interventions. 2, short duration clinical trials, right? That's the other benefit of having a process where the mechanisms in terms of how it's resulting in disease has been highly characterized. The benefit is it gives you short duration clinical trials because of accelerated aging mechanisms. And of course, the third reason is just the vast number of patients out there who are susceptible to it. It's the 9th leading cause of death worldwide. Now, based on all of this, there's potential to have a shorter duration clinical trial focused on an unmet medical need, but also adding exploratory endpoints of aging within that trial, because it's the same process of necrosis that then results in your kidney aging, just at a much faster timeline. And if you ask yourself, why have the regulators, the FDA, EMA, MHRA in the UK, and so on, why have they not approved a drug for aging to date? And there are 2 large hurdles. One of them is a lack of mechanistic insights when it comes to aging more broadly. The great thing about the kidney and necrosis is you have those mechanistic insights. And 2, you're looking at decade-long clinical trials and effects, so prohibitive costs and timelines. So with an accelerated model like the kidney, you're circumventing both these problems. And so the aim there is to get this approved as not just the first drug for kidney injury, but also the first drug for aging.
When it comes to your clinical trials and using your drug with respect to kidney disease, what are you hoping is success in that respect?
Yeah, so in our case, you have gold standard markers of kidney function. For me, success would be showing improvement of kidney function using those gold standard markers. And of course, the other side would be trying to get this approval for aging. And of course, both of these are subject to discussions with the regulators. But again, based on the mechanistic insights we have for necrosis, I am hopeful that there's good potential for both those things.
All right. What are the next steps now for the company?
The next steps are to follow the data. So we are generating vast amounts of data around these drugs, gearing up for those clinical trials. And unfortunately, in science, all you can do is follow that data.
Has AI made any difference in that as you generate more and more data? Is that useful?
Yeah. Our platform is AI enabled. That's how we made the anti-necrotic discovery in the first place. And when you think about how you can use AI for drug discovery and development, there are many different areas. So you can use it once you find a good target, you can use it to ensure the right drug is hitting the target. You can use it to try and improve the success rate of your clinical trial. So yes, of course, we're making use of all of these, but I think the greatest upside, the most valuable, is using it to identify the molecular target in the first place. And for us, certainly that's where the greatest value was, finding the targets to hit and finding the anti-necrotic to begin with.
Are there other companies or researchers doing much work in this field? Because it seems like you're out on your own at the forefront of this.
Yeah, I mean, if I were to give you a list of companies looking at aging using AI platforms, I could give you a large list. In terms of necrosis, this is a first-in-class means to block unprogrammed cell death. we've discovered a fundamental new piece of biology, which is again why something that was attempted for decades we're able to do for the first time in the lab. So again, the effects we're seeing in cell culture, I was really surprised. I mean, they're mind-boggling effects. And we've got some really renowned clinicians and scientists supporting this work, some of which have worked on necrosis since the 1980s, actually. So in that sense, yes, we are the first. And it's been an exciting journey. I mean, we made the discovery in 12 months, so.
I guess when you mentioned about the fact that a lot of what you're doing is very new, what's the approach to that? Because clearly sometimes it sounds almost fantastic when you're coming up with new concepts.
Yeah, absolutely. I mean, I think often when you go into areas where you're really pushing the frontiers of science, you're going into very uncharted territory, then you do get this sense of, well, is that more science fiction? Could one ever actually go there? And the way I've always looked at it is, well, everything seems impossible until somebody does it. And I think necrosis is one of those areas where it has seemed so impossible. How do you intervene in this chaotic, unprogrammed, unregulated process of cell death? And the point is, again, it seems impossible till you find a way. And the way in was the factor modeling. Are there factors within that chaos that is giving rise to the chaos, right? Are there core fundamental factors that you can block such that now you can protect cells to the point where we just couldn't do it before? And often, I think where you have a simple answer, that's where you know the science is actually quite strong. And it's a simple answer that fits with the various disciplines of biology. It explains past findings and it makes new predictions. And certainly what we have with the anti-necrotic does those things.
I'm not suggesting for one moment that you tackle more than you're already tackling, but are there other areas of science that you think, or of human biology and disease, etc., that you think may benefit from a similar approach?
Oh, absolutely. I mean, our drug discovery platform is geared to make more discoveries just like the anti-necrotic. And we already have a few other discoveries in the pipeline. So we've just focused on the anti-necrotic because the potential is so vast. The discovery here is such a fundamental piece of biology, and it has such potential to be game-changing that for the time being, we're focused on it. But the idea is, of course, to develop it and then move to the next discovery.
It's always good to talk to LinkGevity, who I first met at a space event in Glasgow, which is just over an hour from where I live, if there's no traffic, and no temporary traffic lights, both big ifs. Always good to talk about space and the really interesting things LinkGevity is doing around aging and necrosis. A very fascinating subject. Anyway, that's it for another podcast. If you do have questions or comments, suggestions for guests, or want to sponsor one or more podcasts, please do get in touch. And you can do so at jim@deeptechdigest.com. So until next week, wherever in the world you are, I hope you have a great week. Thanks for listening. And I hope you'll join us again next week for another Base to Base Biotech podcast. The Base to Base Biotech podcast is an Air Coastal Media Limited production.